Good afternoon. My name is Julian Ramirez, and I will present my work entitled Molecular Characterization of Population Rhinella mirandaribeiroi and Rhinella granulosa. I would like to start with the following sentence. The first part of knowledge is getting the names right. So, the Rhinella granulosa group is a species complex composed of 13 species widely distributed in South America and Panama. When this species complex is Rhinella mirandaribeiroi. It was described by Gallardo in 1965 for the island of Marajó, Pará, Brazil. Previously, it was known as Bufo granulosus mirandaribeiroi. The orange color represents the distribution of species according to Gallardo at that time, and the blue color shows the distribution of Bufo granulosus lutzi, associated with the Cerrado biome. It is important to mention this distribution because that changed with the work of Narvaez and Rodriguez in 2009, where lutzi was considered synonymous with mirandaribeiroi, and new records were reported in different locations in Brazil and is now known as Rhinella mirandaribeiroi. So, the molecular and phylogenetic analyses carried out for the group of Rhinella granulosa do not include individuals of Rhinella mirandaribeiroi from the type locality or even from those distributed in the southeast of the Cerrado. Therefore, it is necessary to consider them in a new taxonomic revision. That's why we want to answer the next question. Is there interspecific genetic variation between the typotypical population and population distributed in the Cerrado of Rhinella mirandaribeiroi? Also, we want to answer this question. Is there genetic variation between Rhinella mirandaribeiroi and Rhinella granulosa? Because part of my project includes analyzing morphological and acoustic data, and the results found so far do not allow me to differentiate between the two species. So, the genetic analysis was performed using COTS-1 and this and 16S. This will be the first 16S sequence for Rhinella mirandaribeiroi for the type locality. Most of the sequences used in this work were downloaded from GenBank and aligned using MAF and edited with BioEdit. Standard nucleate diversity and haplotype diversity in this were calculated using DNSSP. The haplotype word for the COTS-1 and this 16S datasets were generated using the Apollo viewer taking as input a maximum likelihood topology generated in Mega. A principal component discriminant analysis was performed, also an analysis of molecular variance based on locality by state. Finally, we estimated the species tree for Rhinella mirandaribeiroi and granulosa using the Bayesian-based approach. So, the first part of the results correspond to COTS-1. The default diversity indices for each population locality are shown in the following table, all presenting a single haplotype, which means that there are no nucleotide differences in the sequence we evaluate, despite the large distance between individuals in Rhinella granulosa. Unlike Rhinella granulosa, mirandaribeiroi presents a high diversity of haplotypes, with values of 1 or close to 1, which means that there is a high probability that two randomly sampled individuals are different. In the following figure, we present the generated haplotype networking and the map with the evaluated location on the left. The large orange colored circle represents the only haplotype of Rhinella granulosa, whereas the 10 blue circles represent the haplotype of Rhinella mirandaribeiroi. This gene allows me to distinguish between these two evolutionary lineages, but it presents a high degree of conservation with the population of Rhinella granulosa associated with the Caatinga and Atlantic forest biomes. This is contradictory to what has already been published for the COX-1 gene in a neurons, where it presents a high variation. And now the results for AMOVA for COX-1. Here I am showing the table generated in the Arlequin program when comparing the two species. What we highlight from the table is the value of FST, which is a measure of population differentiation due to genetic structure. In this case, as we obtain a high FST value with significant p-value, we can conclude that there is a genetic differentiation between the evaluated population, or rather, between the species. For Rhinella mirandaribeiroi, a FST of 0.30 was obtained, with a significant p-value, which means that within the localities of this species we found a degree of difference. And Rhinella granulosa obtained an FST of 0 because there is no genetic difference between the individual sample. And finally, the results of DAPC and the majority consensus tree. Both figures are showing me two groups, in blue color for Rhinella mirandaribeiroi and in orange color for granulosa. COX-1 based Bayesian inference tree shows two supporting clades with posterior probability equal to 1, which means these two analyses are also suggesting to me that the individuals I am evaluating with the COX-1 gene belongs to two different lineages. Now, the second part of the results correspond to the 16S ribosomal RNA. The default diversity indices for each biome are shown in the following table. There is a high haplotypic diversity, results that contrast with those observed for the COX-1 gene. A network of 16S genes haplotypes shows two groups. The first in blue representing the haplotype for Rhinella mirandaribeiroi, where we can see that on the map they are found mainly in the Cerrado and the Amazon forest. The second group represents haplotype of Rhinella granulosa, located in the Atlantic forest and Caatinga, shown on the map in red. The yellow arrow indicates individuals from the type locality of mirandaribeiroi, and the green arrow indicates individuals distributed in the Cerrado. In the haplotype network, we observe variation between both populations of mirandaribeiroi. We obtain a FST of 0.82 when comparing both species with a significant p-value. Within Rhinella mirandaribeiroi, a FST of 0.78 was obtained with a not significant p-value. And with Rhinella granulosa, we did not observe population structure either, giving its FST value and not significant p. And finally, the result of the DAPC and the majority consensus tree. Both figures are showing me two groups, in blue color for Rhinella mirandaribeiroi and in orange color for granulosa. A 16S based Bayesian inference tree shows two supporting clades with posterior probability equal to 1, which means these two analyses are also suggesting to me that the individuals I evaluating with the 16S gene belong to two different lineages. So now the conclusion. For the first question, we found no evidence for interspecific variation between the topotypical population and the Cerrado population of Rhinella mirandaribeiroi. Both COX-1 and 16S show interspecific variation within population of the species and showed that there is a structure with the species. And for the second question, we conclude that COX-1 shows population uniformity, a single shared haplotype, while 16S shows more variation. Although there is a clear differentiation at the genetic level between granulosa and mirandaribeiroi, the sample number is small and does not cover the geographic range of both species, so further studies are necessary to validate the taxonomic level of both species. And thank you to the Studying Council for the opportunity to attend and present my work. Thank you ISP, UFU, CAPES, and thank you very much.
Thank you, Julian. That was a really interesting talk. Do we have any questions from the audience? The participants feel free to ask any questions in the Q&A box so that the speakers can answer them. In the meantime, I have a quick question. So in the last slide you mentioned, or the second last slide, you mentioned that you need some future works, future experimentations that can be done to actually understand the distinction between these two species or the similarities between the two. So do you already have any ideas on how you want to proceed from here?
I think that I am not going to do that studies in the future because I am doing my master degree and I don't have time to do that. So I am going to stop there. Maybe if I continue to doing my doctorate, I will do that. But right now, I am going to stop right there.
Okay. But do you have any ideas on how this can be done?
Yeah, we have to carry out sampling through the distribution of both species and in order to clarify the taxonomic status of Rhinella granulosa and Rhinella mirandaribeiroi. So we have just to do that, but we need a lot of time and resource to do that. So it's not easy.
True. Thanks.
You're welcome.
Looks like we don't have any other questions.
Okay.
So thank you once again, Julian.
Thank you.